Alright guys, so welcome to Subu Medicine, and this is Immunology. So today we're gonna discuss about the mechanisms of the innate immune systems. I've already discussed about the general concept of the adaptive and then innate immune systems. I would highly recommend that you watch that first before watching this. Alright, so here we're gonna just go very detailed about the pathophysiology of the innate immune systems. All right, now, if you see right here, right, what do you see right here? So basically, like here, you can think about this as a bone marrow house, and then here you have all these neutrophils, the white blood cells, you have monocytes, basophils, eosinophils, you have NK cells, and then you have B cells and T lymphocytes, right? The importance of this is that, I'm trying to tell you guys the concept of something called margination pull. All right, margination pull. Let me just write down here the concept of this margination pull, which basically means is that, right, these guys, all these white blood cells, they get synthesized in a bone marrow house, and most of these guys go and reserved at your secondary lymphoid organs. All right, so so the synthesis or their primary home, you can think about, is a bone marrow house. Then their secondary home is going to be, look, this is a spleen right here, this is like a pair of patches, lymph nodes you can find there. All right, and this is because they're reserved, they're located around this time. All right, so because of this, okay, we call them, all these cells, they are marginated, right? They're reserved, and you can find them abundantly, okay, present in the bone marrow house and all the secondary lymphoid organs. A lot of times what happens is that, there is these guys from the secondary lymphoid organs, they kind of like, circulate circulate from the circulations and also come back and do home, sit around in the lymphoid organ, all right? And it's really important that you know this because, because what happens is, especially like the bone marrow house, okay, they have this cell called stromal cells, and they provide the nutrients and all for all these WBCs. So they like to be in their home, and their home is obviously this lymphoid organs, all right? So this is the concept of margination pull. Remember, it's a word pull. There is going to be different. There is a difference between marginated versus margination pull, okay? So whenever you think about margination and pull, always think about their reserve. They're located. They're most abundantly found there. All right? Now, that's the general concept of this one, right? Now, you see there's a blood vessels right here, and this is what we're happy to talk about. All right, so now we're going to discuss the innate immune system. So, one of the most important concepts you have to know is that, all right, let's say that, any sort of pathogens or bacteria, any foreign substances that passes through our skins, that passes through your physicals or anatomical or these functional or these chemical barriers, and it get into the tissue, right? It get into our cells. So when that happens, how does our cells respond to the foreign substances? That's what we're discuss about. So what is the most important concept here is you see this like this right here, this pink. This could be a bacteria, okay? We'll just say this could be like a bacteria. We'll just say this is like a bacteria right here, right? And you can say this is let's say this, we can say this is a virus right here. All right? And this say we can say this is a fungi. All right? So basically, all right? This is something that you remember. So let's say any of these bacteria pathogens, okay, any substances, okay, when they come, what is the first thing they're going to do is that, as we said, there are sentinel cells, which are your macrophages, the dendritic cells, natural killer cells, the basophils, right? The main ones from the innate immune system is going to be let's write it down here. The innate immune system is going to be your macrophage, okay, dendritic cells, okay, and then NK cells, also basophil, okay, and these are called sentinel cells. All right? Because these guys are found in the surface of the epithelium, right? So when you have the virus come, what happens is that these are the macrophages, dendritic cells, they are the one that first respond to this foreign substance, this pathogen, this infected agents, right? And one thing that you have to remember here is that, right, this virus, okay, first thing that's going to happen here is that recognitions happen. So let's write down here. The first step is the recognitions, because how do you know, okay, the recognize. So basically you could say recognize happens. The way it works is that the viruses, okay, this bacteria, they show something called, let's write down here. They have something called special, something called PEMP. What is this? Which basically means the pathogen, right down here, associated, okay, the molecular pattern, right? So, you know, what happens that all these guys, they have this pattern that this macrophages can actually recognize that, right? And the way, you know, so this is what your viruses or this is what your, let's say, your bacteria has this, right? The PEM. And let's say this is my macrophages. I'll just make this as my big macrophage right here. All right. So this is my big macrophage. You know, what happens is that, you know, it has, all right, macrophages has something called, let me write down here, something called PRR. What are this? The PR, let me actually erase this right here. Let's write down here. The concept of PR, which basically means it's a pattern recognition receptor. It means, that's what it means, okay? And these pattern recognition receptors are the bunch of them, alright? So let's write down what are they. We have like a lot of this called like tall, okay, like receptors. We call them tall-like receptor. We have tall-like receptor 1, okay? We have tall-like receptor number 2. We have tall-like receptor number 3. We have tall-like receptor number 4. We have tall-like receptor number 5. All right. We have tall-like receptor number 6. We have tall-like receptor number 7. We have tall-like receptor number 8. It goes like that. Okay. We have all the receptors. Not only this, but these are located. Okay. Some of them are located in the plasma membrane. But remember, I'm not asking you to remember all these names of receptors and their each function. But remember, the locations of the receptors are really important. Some of them, like for instance, tall-like receptors 1 and 2, these are usually located in plasma membrane, right? And you know what this tall 1 receptor they can recognize? Okay, let me erase this bacteria right here, okay? This tall-like receptor 1 and 2, you know, they can recognize this, okay? What is this? Lipopeptidase. Okay? In bacteria. Or, you know, that's what it's recognized. And if you see like, let's say, okay, I'll skip this one, but I'll come back to that. And this tall-like receptor, you know, this tall-like receptor 4 can recognize, okay, the gram-negative bacteria. What do they have? They have this lipopolysaccharides. It's called lipopolysaccharides, right? And this tall-like receptor number 5, you know, it can recognize some, like, you know, fungi. All right? Fungi. And this tall-like receptor, even 6, this one, and even the tall-like receptor number 2, it can recognize peptidoglycan. Alright? In bacteria. Alright? And, like for example, this tall-like receptor number 3, what is the importance of? You know, these are found, do you know what they are found? This is a very important concept here. These guys are found in, let's say here, what is it? You can find this is let's say my nucleus right here. Okay? These tall-like receptors are found inside the nucleus right here. Okay, in the DNA, you know, and then what do they do? They have receptors of the tall-like receptors, and these guys are important for like, let's say tall-like receptor number 3 for the double-stranded RNA virus. Like for instance, I'll give you an example, like West Nile virus, or even our recent one, the COVID-19. The coronavirus, okay? These are double-stranded RNA virus. So, look, importance is that. Just know that. Oh, you know where the receptors are located. Some of the receptors are located in the plasma membrane, like for example, tall-like receptor 1, tall-like receptor number 2, tall-like receptor number 4, tall-like receptor number 3, tall-like receptor number 8. Okay, this is for influenza virus, okay? They're located inside the nucleus, okay? And Not only there, okay, there's some of the receptors that are also located in the cytoplasm. Because remember, some of the viruses, right, what happens is that they can go easily, they're trying to find their host. So they can easily come inside the cells, alright? So in that case, there are also receptors located inside the cytoplasm. Like for example, like RNA viruses, alright? Like for example, we'll just write down RNA viruses, okay? And there are other receptors, like for example, mannose receptor, like mannose phosphate receptor. They have scavenger receptor. This is like a receptor I'm giving it to you, okay? There's also another receptor called REG, which is basically means is that retinoic acid inducer gene receptors, okay? There are lecithin receptors, okay? That's for the carbohydrates. So basically, these are all these different receptors, that are found. And these guys are, okay? All the receptors, all of them that I just mentioned right now here, these guys are what? These guys are PRR, which basically means it's a pattern recognition receptors, okay? And who has these receptors, okay? That's something that you remember. Your antigen-presenting, your sentinel cell has this receptor, because these receptors, okay, they are the one they will recognize this pattern, associated molecular patterns in a bacteria or in pathogens or viruses, okay? So these guys are located, remember. So I'll repeat this. You don't have to remember all this name of the receptor. Most importantly, remember, these receptors are located either in the plasma membrane, in our endosomes, or in the cytoplasm. Like for example, and I also give an example for cytoplasm right here, retinoic acid inducer gene. You have a RNA virus, alright? They're located in the cytoplasms here, right? So these are usually like 3 places where you can find, right? Plasma membrane, nucleus, endosomes, or in the cytoplasms, alright? So these are the places where you can find the receptors for these bacteria, viruses, alright? And then fungi or any parasites or any sort of foreign substances, alright? Because these guys, when they recognize, then they're able to do functions, okay? Because any pathogens or any bacteria, viruses, when they get in, the first important thing is that they should able to recognize, that foreign particles, okay? And this is how the recognition occurs, okay? Based on the different receptors, alright? So, after this, alright? After this, I have to erase this part, and then we'll go talk about how this works, alright? So, now, let's say, what we're going to do is that, okay, let's say that, we have this pathogen is coming and attacking us, right? And when they get attacking, what is going to happen is that, look, there is a portion right here, okay? This is a receptor right here. Let's say we can say this is a toll-like receptor number. Let's say I'll just say this is a toll-like receptor number 4 here, alright? And here you can say, like, other receptors too, but let's focus on this one, right? Let's say this is a toll-like receptor number 4, right? It is going to, like recognize this pattern associated molecular pattern, right? So what is going to happen here is that, look, whenever you look at the toll-like receptor, it looks something like this. And these are like leucine rich, okay? Leucine rich. And if you're leucine rich, if you remember the leucines, because one of the phage is facing outside of the plasma membrane, which are leucine rich. And leucine is amino acid that's a hydrophobic, and its domain is facing the inside the cytoplasm, inside the cell, right? Now, what happened is that, okay, when this part right, it can recognize this virus, okay? And it can take it. When it can recognize, what is going to happen? This guy is going to get activated, and then what is going to happen? It is going to give a signal to this domain right here, okay? And this domain, we can call them a toll-like receptor inducers, okay? A recruitment like domain. You can think of the same. You can simply think of that way. This is a domain, right? There's an internal domain that gets stimulated. Okay. Once when this receptor recognizes BAMP. All right. Now, then it gives a signal. Then after that, once this internal domain is activated, okay, the TIR, what is going to happen is that is going to cause different signal transductions, alright? And I'm not going to go super detailed about this because the pathway is very, very complicated. But I do want to let you guys know is that what happens is that, pathway lead into, activations of very, very important, this IKK, which is basically stands for, this is a kinase cascade, okay? So I'll write down here this is called kinase cascade. And reason why I'm mentioning it is because sometimes there's a deficiency on this, right? And then what is going to happen here is that, right, there's a kinase cascade gets activated, right, with a bunch of steps, right? And then what is going to happen is this kinase cascade will go, and there's going to special, let me write down here, special genes, look, special protein is present there, okay, and this protein is associated with something else. What is this? Look, you see the pink color right here, okay? This is called inhibitory kappa B, okay? That's what it is. And what is going to happen? This pink is, okay? And this red right here, this is called nuclear factor, okay? Kappa B. It's really important concept, guys. What happens is that when this IKK, because it's a kinase cascade, what happens? It puts a phosphate group into this. You know what is going to happen to the this I, kappa B? This will go, this is going to go disintegration, meaning that this will break down, okay? And the breakdown will happen based through the ubiquitin pathway, all right? This one. And when this breakdown happens, what is going to happen is, these guys, let's just make this here, all right? So let's just say this breakdowns happens right here, okay? When the breakdowns happens, look, they can no longer get bound with the nuclear factor kappa B, okay? Quick concept here. When this I kappa B was bound or attached with the nuclear factor kappa B, what is happening here is that this nuclear factor kappa is not free to move. It is bound with someone, okay? And remember, nuclear factor kappa B, this one, okay, these guys are specifically, we call them, all right? We call them what? We call them a transcription factor, okay? That's called transcription factor. Very important concept. Now, so when these guys, okay, when the IKK, the kinase cascade pathway, when phosphorylates the I kappa B, this I kappa becomes disintegrated, or let's say detaches from the nuclear factor kappa B. The nuclear factor kappa B, okay, is going to become very, very happy because it is going to do some function. It's going to do some function. When it's free, it's going to go straight to the, okay, we'll just erase this part right here. Okay, we'll erase this part. It is going to go to we'll make this as, okay, it is going to go to let's say, it's a cell right here, and it's going to go to the nucleus right here. This nuclear factor kappa will now, what happens, it will go to the nucleus, inside the nucleus, because this is a transcription factor. And when it goes, what is going to happen here is that it is going to activate the let's say, inflammatory genes, all right? When it activates the pro-inflammatory genes, okay, what is going to happen? The pro-inflammatory genes started, doing the replication, the transcriptions, and the translations, which is going to happen in where? In the endoplasmic reticulums, and ultimately you're making what? You're making the protein. Okay, but then here, what kind of this protein you're making? You're making the pro-inflammatory proteins. And what are those? We call them cytokines. Alright? We call them cytokines. And through this process, okay, what are the cytokines this is producing? Important concept. Here, through this process, okay, I want you guys to remember interleukin number 1, okay, interleukin number 6, and then tumor necrosing factor alpha. It is a very important concept. You have to remember this, guys. What are cytokines? These are cytoreference kines, okay? And there's a movement. Basically, that's what it means, right? So, they produces this cytokines, which is interleukin 1, 6, and tumor necrosing factor alpha, okay? These are very, very important, guys. You have to remember this, alright? This is what's going to cause inflammations. Alright? Now, after this, alright? I'm gonna come back to this. But I'm gonna talk about, this virus too. Let's say this virus got insert, okay? When the virus got insert, remember, what is going to happen is that it is not going to lead into activations of nuclear factor kappa B. Instead, okay, when the virus got into, let's say coronavirus got into, okay, double-stranded RNA, it's a double-stranded RNA virus, right? Remember, who is going to recognize those? Obviously, the Toll-like receptor, which are inside the nucleus, right? But one of the most important things is that when this virus gets into here, okay, let's say coronavirus, what is going to happen is that there is a cascade of pathway that gets activated, but that will activate, okay, I'll just quickly talk about it. It will activate something called interferon regulatory proteins, okay? They will activate something called interferon regulatory receptors, even say that, okay, IRR, let's just write down for our skills. They'll activate the interferon regulatory receptors of proteins, okay? They'll activate that. And that will lead into the activations of the interferon gene in the nucleus, okay? Let's say interferon nucleus in the gene. And from, let's say these are interferon nucleus genes, and this will produce what? They'll produce interferon. They'll produce an interferon, like type 1 interferon, alpha and beta, alright? They'll produce type 1 interferon, alpha and beta. Look, what is the importance of this? I'll tell you a little bit of the interference quickly, and we'll go over that, okay? Look, let's say this is my cell right here, alright? Let's say this is my A cell right here. What happens is, let's say the virus came here. Let's say this virus, okay? It came in and attached the cell. You know what is going to happen in the process? There will be activations of the interferon, like proteins, interferon genes, like interferon regulatory genes and all, right? And during this process, what is going to happen here is that, okay, it comes this activations of those interferon regulatory proteins and genes, and this is what happens is that this will start producing those type 1, let's say interferon, alpha, okay, and then beta. Very important concept, okay? When these guys are produced, right, what is going to happen is that these cells, okay, because the virus is attached, because virus always looking for host cells, right? What happens? This cell is going to die. But then remember, what is going to happen is when the interferons are produced, let's say these are interferons right here, okay? The interferons right here. Okay, I'll make a different color. Like these are interferons right here. These interferons are what is going to happen? They'll go to the adjacent cells. Let's say this is another cell. Let's say this is a B cell. And what is going to happen is that they will go and they will kind of like attach with this another cells. Okay, there's another cells right here. Okay, we'll make another cells. Okay, this is another cell right here, okay? And what is going to happen? They'll attach right here, like this. Okay? So when they are attached with these interferons, what is going to happen is that, it will start making, okay, these cells will start making something called antiviral proteins, which causes this virus not to multiply, okay? So this is how the interferon plays a role, alright? Remember, it's just the activations of nuclear factor kappa does not happen. And usually whenever we talk about interferons are mostly respond to the viruses, alright? So that is why I had to talk about the concept of the interferons, alright? It is a very, very important concept right here, alright? It is a very, very important concept. Alright. So, it's a very important concept here, right? So I had to talk about that. Now, after this, alright? Now let's go back here, right? We're talking about the bacteria. So now, what happened was that we're able to produce these cytokines, interleukin 1, 6, and 2 necrosing factors alpha, right? Now, at the same time, one thing that I had to talk about right here is that, during this process, like Toll-like receptor 4, there are other guys are also helping there, okay? In order for this to gobbled up really nicely. There's one protein called CD14. You can remember this, cluster differentiation. This also amplifies the receptor Toll-like receptor 4 to gobble this pathogens, alright? They're quickly mentioning that too. Now, what is happening here is that once it's produced here, right? The interleukin 6, the necrosing factor. You have to know a couple of concepts here. During this process, right? During this part, because remember, where this is whole thing is what? It's a macrophage. We're talking about this whole cell is a macrophage, right? During the process, what happens is that, macrophages right here. During the process, the macrophages, what it does is that macrophage is sort of like, not only produces interleukin 1, 6, and T1 nucleosine factor alpha, but it also produces during this process, okay, interleukin number 8, alright, and then also like this in the formulated methionine or peptide, you can simply call this as, alright, it produces this, okay, and some complementary proteins. We'll talk about what those proteins are. Because usually complementary proteins are produced by liver. Alright? Now, all of them, okay? These guys are not cytokines. Okay? They're not cytokines, but these are chemoattractants or chemokines. Alright? Chemokines. What different? Cyto is referring as cells, okay, because it's work on the cells, and kines are just kinds are per movement, okay? And these are chemokines, meaning the chemical movement, okay? What it basically means is that this is going to attract, specific, neutrophils to come, okay? This is how it will attract, or it will show directions for the neutrophils. So it's really important. Interleukin number 8 and formulated peptide and complementary proteins. I will talk about what those complementary proteins are too, okay? And you guys have to remember this. Each one of them has a function. We'll talk about it, right? Now, before I go, okay, this unformulated peptide that is obviously coming out from the bacteria, okay? When macrophages gobbles up and do phagocytotic process, it releases this unformulated peptide from the bacteria, okay? And this also play the chemoattractant, alright? And the interleukin 8 is also produced by the macrophages, alright? And we're going to talk about a little bit about the complementary proteins, okay? These complementary proteins are synthesized by the liver. But what is the most important about the complementary protein here is that, okay, let me just write it down, a couple of concepts. What is going to happen with the complementary protein is that one of the most important things you should know about 9 complementary proteins, okay? And these are produced by liver. So we can simply say you have C1, you have a C2, you have C3, you have C4, you have C5, you have C6, you have C7, you have C8, and you have C9. Okay? You have to remember this. These are complementary proteins, okay? What is the most important thing about complementary protein here is that, what is happening is that these complementary proteins, let's just say this is my, C3 proteins, okay? The way it works is that whenever let's say there is a bacteria or pathogen, what happens, this C3, it gets activated and gets split into, let's say C3A and then C3B. Okay? And what is going to happen here is that this C3A, what is going to happen? When this C3A, C3B happens, right? These guys will react into another complement right here. Okay, we'll make this, we'll make it a different color. Let's make it this one. And this one is my C5. Okay? And this C5 will break down into, let's say C5A and then C5B. Alright? And this C5 will, what is going to happen, guys? They will, make this compound. They will activate the complementary from C6 to C9 complementary, okay? And they will make something called all of them get activated, and they will make something called MAC. Okay? So, what is the most important concept here? Remember, whenever there's a bacteria pathogen, right? The complement C3 will react into it, okay? And C3 will split into, and obviously there are enzymes which does it, but we're not going to go to that. Most important for you to remember is that when C3 gets activated, it will break down to C3A and C3B, okay? And then this will lead into activations of the complementary, this number 5. That will, split into C5A and C5E. And then when C5A and C5B gets activated, they will get activated, all this complement from C5 to C9. Some of the book says maybe C6 to C9 too, right? And when they come, whenever all this complementary they're activated, they come as like together and looks like a cylinder, okay? And they will produce something called MAC, okay? This MAC. What is the importance of the MAC? These guys, they do a bacteriolysis, okay? This is a very, very important concept. And do you guys know what the MAC do? They will do, as I said, bacteriolysis, microbe lysis, okay? And this MAC stand for membrane attack complex, okay? This is a membrane attack complex. That's what they do, alright? And, alright? Then after that, what I have to talk about here is that this is how, okay? This is how they'll respond to microbe. And this pathway, which first activated C3A, we call them alternative pathway, okay? But there's other pathway too. Sometimes what happens is that there's also, what is the most important thing? Sometimes what happens here is that instead of like activating C3 first, it'll activate C4 first. And usually you see that whenever there's a mannose binding lecithin pathway, okay? And that time when C4 gets activated, alright, what is going to happen is that C4 will split into, okay, C4A and then C4B, okay? And then again, this will lead into activations of, look, what is going to happen here is that activations of the C2 and then C2A and then C2B will get activated. And then after that, this will activate C3. The common area is always going to be C3. Okay? It's an important concept here. And I'll tell you what is the important concept. Alright? Now, let me just touch only up to this too. Okay? Because there's other one too, which is a classical pathway, and that are usually for the T lymphocytes. Okay? And we'll talk about that when I talk about adaptive immune systems. Alright? But right now, just know that, this is how the complementary proteins, okay, make this membrane attack complex to break down the microbes, alright? To lyse the microbes. One thing that I do want you to remember is that, you see the C3A right here, or like C5A? These are anaphylotoxins. Okay, these are very, very important. These are called anaphylotoxins. Okay? Anaphylotoxins. Because these guys are important for like inflammations. Okay? Very, very important. These guys are for the C3A and C5A. These are for the inflammation. These are what? These are chemoattractant. These are chemoattractant. So these are the complement protein that I was talking about here. C3A, C5A. Alright? Now, other thing, what about this C3B right here or C5B right here? These guys right here, right? This C3B, what does they do? Okay? Let's just go talk about. Let's just say, okay, I'll just make this as my MAC. This is mine. Alright, we'll just say this. This is my macrophages, okay? What happens is that, bacteria are very, very slimy. They're very slimy, okay? They just stick. Let's just say this my bacteria. Whenever, let's say, this macrophages try to eat it, okay? What happens is bacteria sort of like always sleeps it. It also like, it cannot like, it's so slippery, okay? It's so slimy, so it just comes out, okay? So, the importance here is that whenever this complementary C3b is there, let's say when the complementary C3 goes and binds to this like this, what happens here is that, this macrophages, what they can do? They can actually, stick their tongue, okay, and grab this, okay? So so this C3 protein, when they go and attach to this, the macrophages can actually grab it, so that way this bacteria would not able to come out. They would not able to stick out, okay? They would not able to come out from the macrophages mouth, okay? So they are basically coating this bacteria, okay? Because they're coating and helping with the phagocytosis process, okay? We call this guy, we cannot call them we call these guys are opsonizations, okay? These are called opsonizations. So basically the C3b, the C5b, these guys are important for the opsonizations. Okay? They help for the phagocytic process for the macrophage. Okay? It's really important. But whenever you say C3a, C5a, these are for the inflammations. Okay? They are also called chemoattractant factors or chemokines, all right? But don't get confused with cytokines, okay? Cytokines are this interleukin number 1, 6, and tumor necrosing factor alpha, all right? Now, after this little concept of this, guys, all right? Now, what we're going to do here is that, okay, I'm going to erase this, okay? So I talked about a little complementary protein for you guys too, all right? Now, what is going to happen here is that there are a couple of things, okay? So now this C3A, there's C3A, these are C5, okay? We'll just write down here, the C3A, C5A, and then you have obviously you have a C3B and then C5B. Remember when I said this, okay, the C3A and C5A, what do they do? These guys are they're anaphylatoxin, they're for inflammation, right? You know what they do? These C3A, C5A, you know what they can cause? They can go, okay, let's just say this is what it is. Let's just say this is my mast cells right here, okay? What happens, remember, basophils, when they go to tissue, they're called macrophages, right? When this inflammatory products, when they go to the basophils, okay, it causes two things. It causes these granulations of this basophil. And when these guys get stimulated by these proteins, okay, this basophil, like the mast cells, you know, they started producing histamine, right? Histamine is a very, very important one, all right? They'll also produce like serotonin, all right? But histamine is the main one. There's also going to be produced heparin too. But one thing that I do want to remember is histamine. And not only this, guys, in the presence of this, okay, on the presence of this inflammatory chemokines, you know what they do is that, you know, from the membranes of the phospholipids, you know, these guys will stimulate the membranes of this, let's say the mast cells, okay? And the membranes of mast cells are what? They're phospholipids, okay? So what happens is let's say this is a membrane phospholipids, they will stimulate this enzyme called phospholipase A2, and then that will break this down into arachidonic acid, okay? And later it results into productions of leukotrienes B4, which I do want you guys to remember this. There are other going to be, because this arachidonic acid lead into either cyclooxygenases pathway or lipooxygenase pathway. Lipooxygenase, let's write down here. This is called arachidonic acid, okay? Lipooxygenase pathway lead into productions of leukotrienes. Okay, let me write down properly here. Okay, these are called, you can write leukotrienes B4, okay? They're going to be leukotrienes like C4, D4, E4, okay? We're not going to focus on the C, D, E4 because they do have bronchoconstrictions. But I do want you to focus on leukotrienes B4 because leukotrienes B4 is a powerful chemoattractant, all right? And not only this, they'll also run into the cyclooxygen pathway with the COX-1 and COX-2 thing. These are called cyclooxygen pathway and produces a lot of this prostaglandins, okay? Prostaglandins. So PG E series, like prostaglandins E2, if you remember. Look, let's quickly review what we talked about so far. Look, whenever this transcription factor one comes in, right? What happens? It goes to the nucleus and it will activate the pro-inflammatory genes that will produce interleukin number 1, 6, tumor necrosing factor alpha. All right? Now after that, what is going to happen here? Now, once that happens, at the same time, this macrophages also happen is that these macrophages, during the process, they also releases something called interleukin 8 and also this N-formulated peptide or methionine, they also call this, okay? And at the same time, there's also, there's also complementary proteins which get activated, and the way the complementary gets activated is that based on these pathogens, bacteria, whatever it is, right? All this, there's a 3 different pathways. There's alternate pathway, which first activates a complement 3, okay? And then other one is there's a classical pathway and all the lecithin pathway, okay? But ultimate result is that there is a productions of C3A and C5A and C3B and C5B, okay? Whenever you see C3B, these are for opsonizations, and I talked about. And C3A, C5A, these are anaphylatoxins. These are important for what? Inflammations, chemoattractant. And these guys also, what they do is that they help mast cells to break down producing this compound. What is that called? The histamines. Very important. And not only this, in the presence of this chemoattractants or the stimulations on the mast cells, what also happens is that there's a prostaglandins and leukotrienes are also formed, okay? And leukotrienes and prostaglandins are formed based on the membrane phospholipids, okay? And ultimate result is that you have this leukotrienes B4 and then prostaglandins, okay? Now, these are very, very important concept right here. And you guys have to remember all of them. I'm sorry, but you guys have to remember all of them. Look, whenever you guys talk about, okay, and I'm going to tell you what you guys should remember. Always remember these three main products. Interleukin number 1, 6, and tumor necrosis factor alpha. These are cytokines. Always remember the chemokines, interleukin number 8, and formulated peptide, and then complementary protein like C3A, okay, C5b, all of that. And definitely remember the histamine and leukotrienes B4, okay? Now, interleukin 8 and formulated peptide, complementary protein, and leukotrienes B4, these are powerful chemoattractant. Most potent will be the N-formulated peptide, but these are chemoattractant. Again, cytokines are interleukin number 1, 6, and tumor necrosis factor alpha. Whenever you talk chemoattractant, interleukins number 8, N-formulated peptide, obviously complementary proteins, and leukotrienes B4. These are chemoattractants.